All right, so welcome everyone. Thank you for joining us today. In our CGIL seminar, we have a new member, I would say, of CGIL to present for us today. And I think if you noticed today, you're not able to open your microphone or camera. So I will make that available after Ella presents. So we will have that same situation as previously, where after the presentation there will be a moment for questions and comments. So today with us, we have Ella Dodd. She is a current PhD student at the University of Guelph, working under the advisement of Dr. Christine Bays. She received a dual degree in animal science and applied mathematics from the University of Connecticut in May 2020. Ella completed her master's in May 2022 at the University of Connecticut, focusing on quantitative genetics and animal breeding under Dr. Breno Fragomeni. Her master's research focused on genotype by environment interaction and heat stress in swine. Ella's current research interests are fertility traits, sustainability, and mitigating environmental impact on livestock. So thank you again, Ella, and I'll pass it over to you.
Okay, can you see my screen all right?
Yes, it's
okay,
all right.
Perfect. Thank you for my introduction, Ivan. I will be presenting on an implementation of two-trait analysis for heat stress in swine. This is a project that I conducted for my master's at the University of Connecticut, working with Dr. Breno Fragomeni. Traits that alter their expression in correlation with changing environments are said to undergo a genotype by environment interaction. This means that the phenotype expression can shift depending on the climate, housing, management, or time that the data is collected. Traits that are influenced by heat stress are considered to have genotype by environment interaction, which can make evaluation of these traits difficult because these traits can commonly have significant re-ranking of breeding values when the environmental conditions change. We may find in these cases that our best animal under hot conditions may be our worst animal under cold conditions. Heat stress has kind of taken a back burner in terms of animal breeding and selection. We focused on more economically viable production traits. We have implemented some mitigation strategies like sprinklers and fans to limit the effects of heat stress, which can reduce but not eliminate the effects of heat exposure. Early studies on heat stress tested the use of the temperature humidity index as a measure of heat exposure. The THI calculates the effective heat using temperature and relative humidity. There are various weightings on the temperature and relative humidity that can be used to change, which has a greater effect on the actualized heat. Early studies found that there was high correlation with the THI scale studying milk production in dairy cattle. And since then, there have been many studies conducted using THI to evaluate the heat stress effects on milk quality, milk quantity, and fertility traits. There are a number of unique challenges that we run into when studying heat stress. The first is phenotype selection. This step is a problem across all species. We don't measure heat stress directly in animals. We're often looking for phenotypes that have high correlation with heat exposure. There are traits that have a stronger correlation with heat stress, but are not feasible for industry application. An example of this is rectal temperature, which has been shown to represent when an animal is currently under heat stress. However, it's just not feasible to collect rectal temperature on an entire herd to build the data set necessary for this type of analysis. Another obstacle that we run into is using phenotypes that rely on slaughter, like many carcass traits or growth traits. Since these phenotypes require slaughter to collect data, we're not able to collect measurements under high temperatures as well as thermoneutral temperatures. So using carcass traits means we rely heavily on pedigree data to collect phenotypes on the offspring of a sire or dam that have undergone separate conditions. Heat stress is a problem for pretty much all livestock, but swine are disproportionately affected because of their inability to sweat. In response to high temperatures, swine will reduce their food intake and become more inactive, which will lead to reduction in carcass weight, increased mortality, and reduction in nutrient efficiency. These issues in combination will lead to reduced profitability in the industry. There's an estimated $299 million lost each year to heat stress in the US, with $202 million of that economic loss coming from US finishing herds in the swine industry. As we continue to see longer periods of higher temperatures, producers will start to see greater loss to heat stress in their production herds. There has not been much research up until this point on the impacts of heat stress in swine, and in particular, the impacts of heat stress on growth traits in swine. Zumbach et al. Performed a 2-trait random regression analysis of heat stress in crossbred swine in 2008. This study indicated genotype by environment interaction and suggested that it was possible to select for heat stress. A follow-up study was conducted by Fragomeni et al. In 2016, which also utilized a 2-trait random regression analysis, but incorporated genomic information into the calculation using the single-step GBLUT method. This study was able to validate the findings of the Zumbach study and also began a pipeline for genomic selection of heat stress in swine. There are a number of gaps in the literature due to the lack of research when looking at heat stress studies in swine. To date, studies in swine have not evaluated heat stress selection without the use of a random regression. Application of random regression analysis can be a bit difficult because we generally cannot predict THI far enough in advance to make them easily usable. Additionally, there have not been genome-wide association studies in this topic, possibly because of the difficulty of applying a GWAS in a random regression. Finally, there has not been an evaluation of weighting analysis in heat stress selection. We hope to fill these gaps in the literature through this study. The objectives of this study were to evaluate the use of 2-trait analysis without random regression for heat stress selection to make a more usable model for producers, identify any SNP windows that may play a role in heat tolerance or a lack thereof, and finally test the use of SNP weighting as a method for improving our accuracy in prediction. This study will use hot carcass weight as a phenotype to represent heat stress effects. Hot carcass weight refers to the weight of swine after slaughter where the head and organs have been removed. This phenotype is a growth trait and was chosen both because of the high correlation with heat trends and the feasibility of the phenotype. Due to the patterns of weight loss following heat exposure in swine, this growth trait has high correlations with heat stress. Additionally, this phenotype is feasible because it's generally a statistic that's already collected by most swine companies. The data used in this study was provided by Smithfield Genetics and collected at packing plants in North Carolina and Missouri between August 2008 and July 2014 on a commercial cross of a Duroc sire and a dam resulting from a landrace and large white cross. There was genetic overlap between the two farms, with approximately 30% of the sires having progeny in both states. The data was divided into heat stress conditions and non-heat stress conditions, where trait 1 was defined as hot carcass weight under heat stress conditions, and trait 2 was defined as hot carcass weight under non-heat stress conditions. We had approximately 33,000 observations under heat stress conditions, with about 194,000 observations under thermoneutral conditions. Our initial observations prior to analysis showed that heat stress individuals were on average slaughtered 4 days later than non-heat stress individuals, but were on average 5 kilograms lighter than non-heat stress individuals. So despite the delay in slaughter, these individuals were not able to make up their deficit in body weight. Cleaning of the animal data set consisted of removing animals that were missing both parents, any animals with pedigree conflicts, and any phenotype outliers with records above 4 standard deviations from the mean. Quality control on the genotype data removes SNPs with a minor allele frequency less than 0.05, SNPs with call rates less than 0.9, any monomorphic SNPs, and genotypes with a call rate less than 0.9. After cleaning, we had phenotype data on about 227,000 animals in total across both environments, genotype information on about 8,200 animals, pedigree information on about 553,000 animals. We used a 50K SNP chip, and after cleaning, we were left with about 33,500 SNPs. A THI threshold of 78 was used to divide heat stress and non-heat stress conditions. Heat stress conditions were defined as an average THI above 78 for 30 days prior to slaughter, and non-heat stress conditions were defined as an average THI below 78 for 30 days prior to slaughter. This threshold was established in a previous study on a similar data set using a regression analysis. The temperature and relative humidity used to calculate the THI was provided by nearby weather stations. And this THI of 78 ranges from about 25 degrees Celsius with 100% humidity to about 40 degrees Celsius with 0% humidity. The genetic analysis was performed using a 2-trait animal model where Y was the collected phenotype. The fixed effects in the model included the contemporary group, sex, and age at slaughter of the individual. The contemporary group was comprised of the year, week, and farm that the individual was born onto. The random effects included random litter effects, additive genetic effects, and the residual. This equation is also represented in matrix notation, where again, Y is the vector of the phenotypes, X is the incidence matrix of the fixed effects in vector B, W is the incidence matrix of the litter effects in L, Z is the incidence matrix of the random additive genetic effect in A, and E is our random residuals. The variances for the random effects of the 2-trait model were calculated as shown for the random litter effect, random additive genetic effect, and the residuals. For this calculation, we assumed that the additive genetic effect was normally distributed. The single-step GBLUP method was used to incorporate genomic information in this calculation. This method was developed in 2009 by Misdal and Lagara. This method allows for incorporation of both genotyped and non-genotyped individuals into the calculation and allows for the use of more complex models like these multiple trait models. It also allows us to reduce our computational costs a little bit, our time costs a little bit, and provides equal or better accuracy to other methods. The single-step GBLUP method relies on replacing the inverse of the numerator relationship matrix A with the inverse of the relationship matrix H. The relationship matrix H is calculated using the numerator relationship matrix along with the genomic relationship matrix. The G matrix is calculated using the SNP information, weighting values, and gene frequencies in the population. The diagonal matrix D will be manipulated in our weighting analysis. The SNP effects were calculated by back-solving the genomic estimated breeding values using this equation for SNP solutions, where A is our vector with the solutions for the random additive genetic effect. As I mentioned, the weighting analysis will rely on manipulating the diagonal D matrix in this calculation. The original analysis had D equal to the identity matrix with dimensions equal to the number of genotyped individuals. This method provides equal weighting for all SNPs. The first weighting analysis used the quadratic method for estimates of the variance of the individual SNPs calculated as shown. This approach is run iteratively and allows for SNP weights to vary to the extremes. The quadratic method allows for SNPs to explain 0% of the variance, 100% of the variance, or anything in between. The second weighting analysis used the nonlinear A method for estimates of the variance of the individual SNPs. This approach is also run iteratively. However, it has limitations on the variation, so you don't see the extremes that you observe with the quadratic method. For this method, 1.125 was a constant representing the departure from normality, and the exponent was limited at 5 to avoid those extreme values. The accuracy of each of the weighting methods was tested by comparing to the unweighted approach. The breeding value calculated via the unweighted approach was defined as our quote-unquote true breeding value. And the breeding value calculated using each of the weighting methods provided an estimated breeding value. We then calculated the correlation between these two breeding values as a measure of accuracy for each of the methods. The software used for running these analyses are listed here. We use the BLUP F90 suite of programs out of Georgia, which includes ARML F90, BLUP F90, pre-GIS F90, and post-GIS F90. Additionally, we use the R program Gallo to identify any SNP windows or QTLs that may play a role in heat tolerance or a lack thereof. The initial results for the calculation are shown here. The variance components for the random residual, litter, and additive genetic effects were greater in each case under heat stress conditions. Similarly, there was higher heritability estimated under heat stress conditions, with trait 1 observed to be 0.25 and trait 2 observed to be 0.2. The genetic correlation between these two traits was estimated to be 0.63. The results of the genome-wide association study are going to be displayed through Manhattan plots. Our y-axis representing the percent variation ranges from 0% to 1.5%. The x-axis shows chromosomes 1 through 18, not including the sex chromosomes. We used a threshold of 0.8% of the variance in the trait explained to isolate the peaks from the general noise of the Manhattan plot. So for trait 1, representing the hot carcass weight under heat stress conditions, we isolated 2 peaks above that threshold. The first was on chromosome 1, explaining approximately 1.5% of the variance in the trait, and the second on chromosome 14, explaining approximately 1.4% of the variance in the trait. The second Manhattan plot here uses the same axes as well as the same threshold. So for trait 2, representing hot carcass weight in thermoneutral conditions, we were only able to isolate 1 peak above the 0.8% threshold. This peak occurred on chromosome 14, explaining approximately 1% of the variance in the trait. For the study, we calculated a third Manhattan plot to demonstrate the differences and overlaps between our traits. This Manhattan plot was calculated by taking the absolute value of the difference of the results for trait 1 and trait 2. This plot was used to establish the any SNP associations for the ability to resist heat stress conditions, which we called heat tolerance in this study. It was also hoped to simplify the interpretation of the results from 2 Manhattan plots. Using the same axes and same threshold, we isolated 3 peaks that were above the established threshold. The first was found on chromosome 1, explaining just over 1% of the variance in the trait. We identified 2 on chromosome 14, explaining 1.4% and 1% of the variance in the trait, respectively. The results of the weighting analysis are shown here. The y-axis of this graph represents the accuracy of the method ranging from 0.3 to 0.55, while the x-axis represents the iteration of the method. The traits are going to be differentiated by color, and the method used is differentiated by line type. The nonlinear A method showed the highest degree of accuracy at iteration 1, with a quick convergence in accuracy as SNP weights were allowed to vary. The quadratic method also showed highest degree of accuracy at iteration 1. However, there was a steeper decrease in accuracy following iteration 1 as the SNP weights were allowed to vary. One of the primary outcomes of this study was the conclusion that hot carcass weight under heat stress and hot carcass weight not under heat stress can be considered separate conditions, separate traits. The first indication of this was the difference in heritability for the two traits. We observed higher heritability under heat stress conditions, which may indicate higher genetic variation at high temperatures. This would likely be attributed to the lack of selection that has occurred in this trait historically. Higher heritability under heat stress is also a pattern that was found in the two previous studies that I mentioned, Zumbach et al. And Fragameni et al. Along with our heritability results, the genetic correlation indicated separate traits. The estimated genetic correlation between these traits was 0.63. Genetic correlation less than 0.7 is evidence that the two traits being studied should be considered a single trait, or should be considered separate traits, because analysis of them as a single trait would lead to significant re-ranking in our breeding values. Additionally, this value is a widely accepted threshold for concluding genotype by environment interaction. Our second conclusion of this study was that both traits, along with the defined trait of heat tolerance, are highly polygenic. Our first indication of this conclusion was our Manhattan plots. In all three cases, we were only able to isolate between 1 and 3 peaks, explaining more than 0.8% of the variance in the trait, with the majority of our SNPs falling below this threshold. The highest percent explained was found to just be 1.5% of the variance, which is relatively low. We were not able to isolate a clear few genes explaining all of the variance, which indicates that there is a high degree of complexity in this trait. These results have not been demonstrated in other swine studies. However, studies in dairy cattle using rectal temperature as a phenotype found similar results and concluded the trait to be highly polygenic. This conclusion was supported by our results from the weighting study, which found decreased accuracy in all cases when the SNP weights were allowed to vary. The quadratic weighting method is thought to only improve accuracy in cases where there are just a few genes affecting a trait, which does not reflect what we found here. In contrast, the nonlinear A weighting method is thought to find stability and accuracy in complex traits, which does mirror what we found in this study. Due to the decrease in accuracy for both weighting approaches, this study does not recommend the use of weights in analysis, especially given the added complexities of recalculating the gene matrix as the weights change. The genome-wide association study isolated 6 possible candidate genes that may play a role in heat tolerance. None of these genes isolated have a direct link to heat tolerance. However, some have more complicated relationships with heat tolerance that can be presumed. For example, PRRSV refers to a swine respiratory virus, and resistance to infection under stressful conditions may support growth at high temperatures. However, as I mentioned, these genes do not have a well-documented link to heat stress as far as we understand the biological mechanisms today. There may be more studies into how these proteins may relate to heat stress. However, the regions isolated may be an artifact of the small number of independent chromosome segments. In the swine population, there are approximately 577 independent chromosome segments. Independent chromosome segments refer to the portions of the chromosomes that segregate independently in the population. And this value is directly related to the effective population size, which can be small in domesticated farm animals due to the degree of selection that has occurred in those populations, and the length of the chromosome. This small of a number would cause us to run into an issue of multicollinearity, which essentially means we're trying to predict too many things with too little information. This would increase our predictor error variance of the SNP effects and possibly lead to an instability of our GWAS results. This would mean that the peaks isolated in the GWAS may change drastically, even with slight changes to our data set. Additionally, there is just an overwhelming lack of annotation in the swine genome, which limits our GWAS results. It's possible that more genes will be linked to these regions, or regulation genes may be linked to these regions that have a more direct relationship to heat tolerance as we learn more about the swine genome. The use of a GWAS may help us to understand the underlying genetics, but it is not useful for application in animal breeding when we're dealing with traits that are this complex. For this reason, we do recommend a whole genome approach when implementing a heat tolerance program in swine. The overall conclusions of this study indicate that hot carcass weight under heat stress and hot carcass weight under thermoneutral conditions are separate traits. Additionally, we were able to recommend the use of a 2-trait, single-step, and unweighted approach for implementing heat tolerance selection in swine. Finally, we found that both traits were highly polygenic in nature, with a recommendation of a whole genome approach in these traits. There are 2 primary options for industry application in this study, the first being selection for one trait over the other. This would allow producers to select the trait more relevant to their environmental conditions in their herd. The second would be a breeding program that selects for traits depending on the expected season of slaughter, so changing the preferential breeders depending on when the planned slaughter of the litter would be. I wanted to thank my collaborators, including my advisor, Dr. Breno Fragameni, and Smithfield Genetics for support and providing me the data to work with. And I wanted to say thank you for listening to my presentation. I know this wasn't exactly the presentation you were expecting this week, but I appreciate the opportunity to speak a little bit about my research.
Yeah, thank you very much. That's a good presentation, and yeah, thank you for providing some feedback on your history coming here now to Guelph. Now we are aware of the things that you have done before. And so now I will allow people in the audience, if you have any question or comment, you will be able to do so. You can turn your camera on and unmute yourself, or you can also use the chat, and I can read the question to Ella regarding what she presented.
Ivan,
Yes,
I have a question.
Okay.
I have one volunteer.
Okay, Flavio, and then
yeah.
Christine.
Okay, yeah. Ella, I have a question for you. At the very end, you mentioned the conclusions that basically the heat stress conditions were met in your study based on season, right? So probably you had like the summer season that where the animals were over the threshold.
Yes.
But how about you have more than one location, right?
Yes.
So for the locations that you had, the locations would not contribute to the animals to be above the threshold, the heat stress,
So
because that has implications on the conclusion of your study, right? Because also you could, because the United States, they have a very hot regions in more colder regions, right? So in the United States, because of this variation, you could think about selecting animals for thermotolerance based on the region where they're going to be produced, right? Other than the season, as you mentioned. Can you comment on this? Because I couldn't get exactly the region.
So I think the first part of your question was, would there not be an effective management between the two farms?
No.
Okay.
Related to the location, the region
Okay.
of the farm. So
just the
location as the main driver of the heat stress compared with the season as the main driver.
So we used the same threshold for all individuals in the study. So we had a THI of 78 separating hot and cold conditions. So that was the same across both. There would be differences in the number of days that you would have at those two farms above that threshold. So that would change depending on location, but the threshold itself would stay the same.
Yeah, and that I understood. I'm like comment on the fact that about your conclusion, because you said now this knowledge allows to select for animals that are better for a season.
Yeah,
I guess the hot season, but generally speaking, it's more like you are interested to see if there is regions of the country that would have a hotter seasons or longer periods. So you could select for that specific region.
Yes, you
Right.
could have region by region selection criteria for your individuals. One of the applications that you could use for the study is if you are a producer in a region that has very little heat stress risk, you have very few days of the year that you're above that THI, then you may not want to prioritize that in your selection. So you can basically prioritize the individuals that are better under cooler conditions. However, there's a lot of regions, especially in the United States, that have both. They have very cool, specifically Missouri has very cold winters and very hot summers. And you could also adjust your breeding program depending on when the litter outcome of your breeding would come to slaughter at about like 180 days.
Yeah, I agree. My point is a combination of location and the seasons.
Yeah.
Right, how the behavior of the seasons. Because you just mentioned season, I was trying to think why you did that, why you emphasize just season, because season depends where at the location, right?
Yeah.
That's good. And very nice presentation, Ella, and congratulations for this study. I have one more question, just clarification. In the Manhattan plots, you said the threshold was 0.8% of variation of the trait. Do you mean phenotypic variation or phenotypic variance? Because you always say the variance of the trait. So,
yes.
So you use phenotypic. So is 0.8% of the phenotypic variance? Yeah, that's what you use?
Yes.
Okay. So you usually look more at how much you spend on the genetic variance, right?
Yeah.
So we can maybe talk another day why you decide to do that. So I'm done. Christian.
Okay, great questions, Flavio. My questions are not as theoretical, Ella. I think you did a fantastic job. I don't think you have to apologize to anyone. I think everybody who's still here really wants to hear what you have to say about your study, and you absolutely did a fantastic and very clear job of telling us what exactly you did. I would be interested in knowing, so when we do this type of analysis in dairy cattle, oftentimes we sort of tack on at the end graphics showing the re-ranking of the males. Did you look at all at that, and did you see any real re-ranking between the individuals that were undergoing heat stress and those that weren't?
We did. I didn't print the results in the study. I don't remember the exact percent shift, but there was an adjustment in the ranking of the breeding values between the two traits.
Okay. Yeah, I would expect yeah, I'd expect that, but it's always neat to kind of see how much we're throwing the rank lists off by doing that. But fantastic job. I'm going to stop asking questions. I think that's wonderful. Thank you so much.
All right, so we have 2 questions in the chat. I don't know if you are able to see them. If yes, just let me know. Otherwise, I can read. Or I can read actually. So there's one question from Thomas Strobel. In case one trait switches to 2 traits and genetic correlation between is low, is there any alternative to run 2 separate genetic evaluations and having 2 breeding goals as suggested?
So you do have the option if you wanted to kind of run a single trait analysis only involving the conditions of one or the other. And you could run that if you have different breeding goals in mind, as I mentioned, with like if you're in a region that has only heat stress, you have no cold days of the year, you're going to prioritize heat stress. If you have regions that have very little heat stress, you may not prioritize it as much. So you could run separate evaluations depending on your goals.
Okay, and there is another question from Lucas Lopez. Did you or your lab get to look at heat shock proteins from these animals?
No, we didn't look at them specifically. We were hoping that they were going to flag on the GWAS, but we weren't able to find any association with them in that in the regions that we isolated.
Does anyone else have any questions or comments to Ella? So Ella, I have a question. As you know, I've been working on heat stress on dairy cattle, and one of the things that I've always interested in how to infer or to study heat stress, how to assess heat stress in large scale, and we have so often used the THI, an environmental indicator, and to me, it was interesting to see that you used a THI that was developed and used in a study for dairy cattle, right? And I think I've seen some different indices for swine specifically. So I would like to, yeah, if you would give some comments on how, why you decided to go with this this THI, and also, I guess another thing, just so 2 questions. One is about the index, and the other question is about the threshold. How did they find? Was it like a study that they worked with a chamber and therefore they simulated a heat stress event or not for the swine? So yeah, this is 2 questions.
So they're kind of answered by the same answer. We used a previous study that worked with a very similar data set. So it was not an experimental data set. They ran a regression analysis on the changes in hog carcass weight in response to the THI in the days preceding slaughter. So looking at regions where we would see significant deficits in hog carcass weight. So it was just played with a couple different THIs to find the best region that explained most of the deficit in hog carcass weight that we would see. So both of those, the equation that we used and the threshold was from a previous study in swine.
Okay. And then the question, it's connected to where the animals were in. So did you have information on if it was like an indoor setting and if there was any sort of modification or controlling of the environment? I know swine is very intensive. I'm not so sure in the US, I guess, but in Brazil it is, and they have even some systems that they work with pipes and they go specifically, like, are directed to a region in the animal to cool the animal and all of that. So I was wondering how I mean, that would affect a lot what you see in terms of the environment conditions if you were working with weather station as you did to what is inside and how the animal is actually perceiving that temperature. So how did you, yeah, your thoughts on that, I guess.
Yeah, so we tested differences between weather stations at the farm and differences with weather stations at nearby weather stations. So we first ran that analysis and we found a correlation of 0.99. So we assumed we could use the weather stations, which had a bit more information than the ones on the farm. But for inside of the barn, they had the same management technique. So they both used fans and both were indoor only facilities. So I would presume that there is an impact of the fans on the feeling of heat by the swine. But we are hoping that it would be controlled for by having the same environmental conditions between the two farms. Does that make sense?
Yes, yes, it does. Yeah, thank you. So does anyone else have a question or comment?
I have another question.
Go ahead, Dodd.
Because this was a great presentation, Ella, but a lot of information in a short period of time. So I don't know if I fully understood what you are presenting. The Manhattan plots are the vines explained by windows.
Yes.
Yeah. So how many markers in a window, or it was based on not number of SNPs but level of LD? Can you explain a little bit more about the windows?
So we used moving windows containing 20 markers.
20 SNPs. Okay.
Yeah.
Okay. That's what we started. So that's the variation of the window.
Yes.
And I don't know when you did this study, maybe was not available yet, but I'm going to just ask you if you thought about this, if it was available. If you have looked at the approximated p-value of each marker effect.
We did not look at p-values of the marker effect. We had thought about running a follow-up on the study, but I ran out of time in my master's.
Okay. That would be something interesting to do too, because then you could control better for the number of hypotheses that you are testing, right?
Mm-hmm.
To have more confidence on. But I think you with this the criterion that you use, 0.8 of the phenotypic device already was quite strict. So you just got really, really large effects, right? But maybe you missed some effects that will be significant, and maybe you would find the genes, the heat shock genes that you missed, that you expected, right? Because they were not so large in terms of the variance that is explained. And another thing that you would miss here, if these SNPs associated with these genes are in low frequency in your population, right? So just that would not allow you to explain a lot of variation, right? So, but anyway, just to understand. I think I should read the paper, then I will get all the details. Okay, thanks, Ella.
No problem. Thank you.
All right, so Ella, I think we don't have any more questions. So once again, thank you very much for presenting. It was a really great presentation, and also you raised some questions and discussion. So thank you all for joining today with us, and I hope to see you all next week. Have a very good Friday. Bye-bye.